Okay. 5:45? Oh, we have photo session before. Okay. Yeah, just check, just check, just check. Yeah. 5:45. Okay, fine. So we are back here for this edition of the Wednesday colloquium. So before I start the colloquium, I would like to say that this colloquium is actually, we are very glad that is part of the Department of Chemical Sciences annual meeting, which is called as the TIFR Annual Chemistry Conference, which brings home to TIFR people not only within the Department of Chemical Sciences, but also pulls in people from various other departments and institutes that are prominently doing chemistry or pursuing chemical sciences or associated areas, like in NCBS. We have visitors from NCBS, from TIFR Hyderabad, and it makes it a completely rounded meeting, allows for us to enjoy the chemistry that is going on across different institutes under the TIFR umbrella. So I would like to thank the department chair, Deepa Kushalani, and all associated members, especially Dr. Kalaivanan Nagarajan, for organizing this entire meeting and getting people here. So thanks, Kalaivanan. So coming back to this particular colloquium, I am very happy to welcome Professor Mahalakshmi from IISER Bhopal. And this is actually a pretty nice coincidence that we have her just after she recently won the Rashtriya Vigyan Puraskar, the Yuva Shanti Swarup Bhatnagar Award. And we are very glad that Mahalakshmi agreed to come and be part of this annual meeting. So just to give you a little bit of a background of Professor Mahalakshmi. So Mahalakshmi actually did her bachelor's from Bharatiya University in Coimbatore, and then she moved as an integrated master student to Indian Institute of Science, Bangalore. And this is where, kind of what I heard today at lunch, she had a variety of different training. And the immediate thing that she did after joining in 2000 in this program was to work in a lab where she could look at patch clamp methods. And then she moved from that project to another project which involved computational biophysics, and a little bit of understanding, peptide structures and different kinds of motifs. And then what she settled in finally was to do an experimental work with Professor Balram under the influence of doing NMR, that she wanted to really pursue NMR. And this is the time when Balram was doing starting up a lot of mass spec studies on proteins and peptides. I think Koti remembers one of the talks that Balram gave in this auditorium in 1999 when he came, he started doing that. So, so at that time she joined and she settled in. She looked at peptides which had this wonderful amino acid tryptophan that allowed for an optical measurement of all sorts of properties which so many students in our department use. Has been historically a very significant amino acid in all fluorogenic probes. So yeah, and then she went on to look at different constructs of peptides. And because of her love for NMR, she went into doing a postdoc in UCSD, where she worked in a lab because she told me that she wanted to be slightly in a different sort of measurement scheme if she could use NMR for different classes of measurements. So this was looking at proteins in bilayers without actually, spinning the NMR cell. Okay. So people know that if you spin the NMR cell, you have these solid state NMR can be done because dipolar interactions can completely averaged out. But this particular lab that she worked in, you could do static cell NMR, which is very interesting. And she went into the depths of that. And then not just, the thirst for knowledge doesn't stop people from looking at all sorts of techniques. She went into an X-ray crystallography group. So with two postdocs, she returned back. She came back to IISER Bhopal in 2009. And since 2021, she's a full professor there. And she has won, of course, numerous awards, the Swarna Jayanti and recently the Bhatnagar, but many other awards. But what is wonderful is she came out of what she was doing. She took the lessons from her postdoc and PhD and came up with completely new set of science that she's pursuing in IISER Bhopal, which started because of a paucity of a class of NMR equipment that was not available at that time in Bhopal. So, and she has been extremely successful at looking at these membrane chaperone proteins. So, Mahalakshmi, without any further ado, please, you can come and deliver the colloquium.
Thank you, Jyotishma, and thank you, Koti, for inviting me here. It's an honor to actually deliver this Wednesday colloquium, and the coincidence that it is also a part of the TACC is an additional honor for me, and I'm so glad that there are so many of you working at the interface, addressing science in very different aspects, all of you enthusiastic enough to leave the very interesting coffee session and come here for the talk. Thank you once again for having me here. So, as Jyotishman actually explained, doing science is an honor in itself. One doesn't expect awards, one doesn't expect recognition, because the biggest award and the recognition is the fun in solving a problem. So one of the problems that we wanted to solve was, how does a membrane protein actually fold? And is Anfinsen's hypothesis really true for a membrane protein? So with this, I'll be talking about a membrane chaperone, which chaperones itself, and how the folding stability and function interrelate in terms of this particular protein. So just to give you an introduction, the cell that every one of us carry in our body, the cell is surrounded by a structure that keeps the cell together. Just like how the walls enclose this particular room and keep it together, the membrane is the structure that holds the cell and its constituents together. Whether it is an animal cell or a plant cell or a bacterial cell, the membrane is what keeps everything inside and keeps it contained. Now, in order for this room to communicate with the exterior, we have what are called as doors. And those doors are your membrane protein. So even if it is a virus that you're looking at, say for instance, the animal virus, a famous or infamous coronavirus, or its distant cousin, the rhinovirus, which causes the common cold, they all have membranes. The only pseudo-organism that doesn't have a specific membrane is a virus that attacks bacteria. Except for that, every one of us have a cell with a membrane. So these proteins that sit in the membrane are the ones that help with the communication process. So what exactly is the membrane made of? So the membrane is made up of molecules which we call lipids, and the lipids have an amphipathic nature, or you can even call it an amphiphilic nature. See, it has a polar region which helps it sandwich with the water outside, and it has a hydrophobic region which helps it sandwich with itself. And two such regions with a symmetry in between now gives you a membrane bilayer, which forms the walls of the cell. Now, this membrane bilayer is relatively impermeable, and because of that, you now need to have molecules that help it selectively permeabilize itself to reagents that are required and maybe not required. So those are done by these molecules called as proteins. And unlike the other proteins, the workhorses that you see in every part of the cell, these are specific because they are anchored to the membrane. They are attached to the membrane themselves. So they are called as membrane proteins. And these proteins constitute 30% of the overall cellular content. Now, if you look at what they do a wide variety of things, whether it is enzyme catalysis, cell-cell communication, or even help establish the infrastructure, that is, the cytoskeleton of the cell. It carries out a wide array of functions that any other biomolecule is known to do. So for these proteins, studying them, people have tried it over the past several decades. And what we have concluded so far is when you look at those structures, you classify them into only two categories. There is no mixture in terms of what sits in the membrane. So they are all helical, that is, they have these ribbon-like backbone, or they are all beta strands, which have a basket-like backbone. So these are the only two categories that you can put membrane proteins into. And the reason they are only in these two categories, the most fundamental reason, is because these two help the protein sit stably in the membrane. So why does it have to sit stably in the membrane, and what are the hindrances that it encounters? The lateral bilayer pressure, that is, the membrane itself sees this protein as a foreign object and wants to push it away. Whereas the protein, if it goes outside, it cannot survive in a water-rich environment because it has got water-repelling residues. So in order for it to maintain a balance, there is a lateral pressure that pushes the protein molecule together, and therefore multiple proteins now try to crowd together and form what are called as islands that float in the sea of lipids. So these islands, which are enriched with proteins and also have some lipids in them, those are the ones that act as hubs for several signaling processes that happen within the cell. So when it comes to helical proteins, how do they fold? They fold by a process which involves their synthesis and their folding going hand in hand. That is, you have the ribosome, which makes the protein. It's the seat of protein synthesis, and it allows the polypeptide to fold into the membrane as it is being made. And there are other proteins, there are translocons, that is, proteins that translocate substances, those that sit in the membrane and allow a new protein to come in. So all helical proteins are made in somewhat this fashion. But when you look at the other category of structures, that is your beta barrel membrane proteins, they are not made this way. These proteins cannot be folded as they are made. And there is a specific reason for that. But before we get to it, where do we find these beta barrels? Now, the helical proteins, we find it at the edge of the cell. We find it in every membrane that is inside the cell. So it is kind of abundant, ubiquitously present. But the beta barrel membrane proteins, they are specific to one specific organelle that is there in all of our bodies, and that is the mitochondrion. Mitochondrion is the site of ATP synthesis, and that is what is giving me the energy to stand here and talk. It's giving you the energy to listen and to also think and to do pretty much everything that any human being is capable of. And the same thing applies for all organisms in the animal kingdom. The mitochondrion is the one that helps it do the job. And the proteins that sit in this outside are beta barrels. Why is it that only in this specific case we have beta barrels? We don't see it anywhere else in the cell. That's because mitochondria came from friendly bacteria 1 billion years ago. So these bacteria, the gram-negative bacteria, diderms, that is, they have two derma, two membranes, those are the ones which have beta barrels in their outer membrane. And that's where we also obtain mitochondria with beta barrels in the outer membrane. So these are the two major places where you see the beta barrels. Now, these beta barrels, they perform several functions, and it's important for us to study them because we all are aware of antimicrobial resistance, antibiotic resistance. And these beta barrels that are sitting in the outer membrane of pathogenic bacteria are the ones which are essential to push these antimicrobials that we give away so that the bacterium survives. So if we identify a lot more about how these proteins function, how they fold, how they are stabilized, then maybe we will get smarter and we'll be able to get better peptide-based or other novel antibiotics. Small molecule, large molecule, doesn't matter, but we'll be able to target them using an information-based design. So for this purpose, we need to study these beta barrel proteins that are sitting in the outer membrane. Now, these beta barrels, what do we know so far? They are a cylinder-like structure. So you take a piece of paper and you fold it, you get a cylinder. The beta barrel does exactly the same thing. So it has the first and the last strand which come together to form a cylinder. Now, based on how many strands there are, that is, how wide your paper is, you will get a cylinder that is small or you would get a cylinder that is big. So, which determines the size of the barrel over here is the number of strands that are there. So if you have a minimum is 8, so if you have 8 strands, then it forms the smallest cylinder that is known so far. And the largest cylinder can go up to 32 strands, and it can form a hole that is big enough to even move other protein molecules. So these beta barrels that we know of have their first and last strands come together through hydrogen bonding. They form cylinders which anchor the protein to the membrane, and the outside region is the business end of the molecule, which is either involved in attaching to the host, invading the host, or even getting in nutrients for the bacterium or for several other functions. This is what we know. Now, how do these proteins get there? Because they are made here. This is inside the cell, inside the bacterium. So it has to cross two membranes in order to get there, and then it has to fold there. So the way it is done is it is moved through these secretion pores, the sec translocon that takes the polypeptide outside, and then it is carried by this molecule which holds the polypeptide in its unfolded state. It's called a holdase. One of the holdases is SIR-A. And what it does is it hands over the polypeptide to this barrel assembly machinery. Yeah, sure.
How does it garner? I mean, what kind of experiments actually showed that, it is going through this unfolded pathway and then, so is it X-ray crystallography or what experiment?
No, these are all biophysical experiments. So the way it was done is people were finally able to identify and extract out these complexes and give it an unfolded polypeptide. They were able to identify that these proteins were able to hold it, and that complex, they were able to run it through NMR spectroscopy and get the structure of this molecule. And then when they added the polypeptide to it, the polypeptide did not contribute to resonances that corresponded to a folded protein. It kept it in the unfolded state. Then they did cross-linking experiments. They did protease experiments. So proteases would digest anything that is not structured and compact. So with all of these, they were able to show that here it's kept in the unfolded state, and then it's handed over. Again, cross-linking, FRET, and other similar experiments were what were primarily done. And then the polypeptide that was given to this was folded. But now we have cryo-EM snapshots of all of them. Yeah.
And they're unfolded. Why don't they aggregate while they travel from this to that end? What is that special about this survey that it
prevents the aggregation of these big unfolded proteins?
So what surrey does is it has both hydrophobic regions as well as charged residues. So what it does is it forms interactions and holds it in such a way that the polypeptide does not get a chance to interact with itself. Instead, it interacts primarily with surrey. And that's how surrey keeps it in the unfolded state, prevents it from falling into an aggregated state. Aggregation, dimerization, oligomerization can happen. There is another partner protein with this called as DEGP. That is an acronym for degradase. Its job is to degrade. So there is a competition between this protein holding it and preserving it and DEGP holding it and degrading it. So most 60 to 70% of what is made is degraded. Only 30% is actually folded. You would consider that that is a wasteful cycle because 60% is degraded and wasted. But the other extreme is, like you asked, what if it aggregates? So to prevent aggregation, the cell goes through this wasteful cycle of degrading it instead.
Could you mention the typical dimensions of these? Like, for example, you mentioned 8 strands and 32 strands in the previous. So each strand would be like what? How many micrometers?
To 15 amino acids. So the span would be about 40 angstroms.
Okay.
Give or take. Of course, that depends, varies based on the lipid, but an average of about 40 angstroms.
We will let her proceed and then
Oh, no,
after that.
feel-
Yeah.
feel free
But
to
at
just-
least, yeah, we want you to speak early. So, go ahead.
So this is what we had known at that time. And when we started all of this work, I was very interested. So 15 years ago, when I joined IISER Bhopal, I wanted to look at proteins, membrane proteins, that were channels, but were not helices. So they were functionally the same as any of the other channels, but they were structurally distinct. Part of it was probably because I have a maverick-like nature. I like to think, not follow the mainstream. So it was my rebellious way of looking at structures, but eventually it did pay off. So these structures that were solved later revealed that this particular barrel assembly machinery is made up of a protein that is a core entity called as barrel assembly machinery A, BAM-A. Then it has auxiliary helpers which sit with BAM-A and help it do its job. So the BAM-A, if you look at it top down, you find that there is a hole that is formed inside it, which allows this unfolded polypeptide to thread its way through. And this hole formation is actually facilitated by this extramembranous, that is the outside membrane region of the same protein, which is called as the polypeptide transport associated or POTRA domain. So POTRA is, there are 5 of them, and there is a movement that happens between some, a movement that does not happen between some others, and this decides whether or not it's going to be dynamic or static. So this information started coming in, and what BAM-A does in the membrane is it is an asymmetric barrel, that is one side, so an average of 25 angstrom for the bacterium. So one side is fatter, one side is thinner. What happens when an asymmetric barrel sits in a solution, a lipid solution? It pinches the membrane on one side. So because of which there is a local distortion or a local chaos that is introduced in this particular system. This local chaos now allows this chaperone to push in a new polypeptide. Otherwise, if it is something that is so rigid and brick-like, it's very difficult to put in something new. But if it is spongy and if it is fluid, then it's easier to insert something new. And that's one thing that BAM-A does just by virtue of its asymmetric structure. The other thing that it does is through its differential flexibility, it creates what is called as a worm-like movement. So extended and compressed, extended, compressed. So this worm-like movement, now if you were to hold a polypeptide in this place, The worm-like movement will have the polypeptide stretched, compressed, push, then go and grab the next one, push, and push it into the membrane. So this worm-like movement allows the potra to fold the polypeptide. The third thing that BAME has is it is a 16-stranded structure. The first and the last strands do come together, but they are not perfect in their arrangement. There is a gap that is created because the first and the last are not perfectly aligned and form perfect hydrogen bonds. That doesn't happen. Because of that, it allows this structure to actually do a dynamic breathing movement in the membrane. And the final thing is this domain movement that is there outside the membrane can either cap the barrel, as in close the lid, or open the lid. So this movement also allows the protein to decide when it should fold a polypeptide and when it should stay in the inactive state. So we had a lot of this. Yes.
No, so how much of this stuff is just known from structure and how much of this is actual some energetics, thermodynamics,
All
sort of thing?
Them. So what we have so far comes from structural studies, that is NMR, as well as crystallography studies of BAME itself, with and without its potra, with and without its auxiliary subunits. It also has all atom MD as well as coarse grain MD studies. Then it additionally has cross-linking experiments that were done to capture all of the dynamics.
So you know you know what's happening to the polypeptide chain as it go. Meaning it seems like the energetics is off here somewhere.
So the energetics is definitely off because it destabilizes the system.
Right.
When it
So
is
you're getting
functional, it
energy
destabilizes.
from somewhere, right?
That's the catch. This system works without any additional ATP. And there is no membrane potential.
No, I
for
know,
it
but
to
there's
help either.
some other place in the membrane where there's something, some ATP which is driving and making the membrane nicer or whatever. No?
No, not in the outer membrane. There is no ATP available there.
So it's just that the protein is really, really unhappy being on the other side, and so it just gets through, and so it gets more stabilized.
Something like that.
Okay.
We're still trying to solve it because we still don't, like you said, the energetics is off, the picture is incomplete. We still don't know exactly what is happening and how it is being driven in the outer membrane.
Okay, thanks.
Sorry, this might be a stupid question, but
There are no stupid
if
questions.
ATP is not giving the energy, I mean, for the thing to move and do some work, some energy has to be expended by physics.
Yes.
So where does that come from?
It comes from the intrinsic dynamics of the system itself.
I see. So
Like Sachi asked, some things, when they are interacting, they stabilize the system. But this protein is deliberately kept intrinsically destabilized. And the membrane plays a role in it. The membrane interaction itself drives this process. Or at least that's what we found so far. So there were several mechanisms, models that were proposed. One of them is that the gate opens, the polypeptide comes in, and then it is released into the membrane. This was a threading model. The other model that was proposed is that it is folded a little bit over here. The whole structure is assembled one hairpin at a time, and then it is inserted. The third model that came out was that it is not folded in the membrane. The nascent polypeptide that is coming in is folded outside the membrane and it swings in and is then released. The fourth one was it doesn't happen outside the protein, it happens inside the protein. That was a lumen catalyzed model. So there were 4 models at least that have been proposed. Some of these substantiated with cryo-EM studies, but there are other models which are equally valid because they also have cryo-EM studies confirming it. So with all of this chaos going on, we decided to ask a few questions on how does BAME itself fold? Because that's also a beta barrel that also has to fold and go into the membrane. How does that happen? If that doesn't fold, none of the other beta barrels would fold. So the fundamental question of how BAM-A itself folds was not asked. So this is your BAM-A. And a recent study showed that when a polypeptide is actually presented to it, this particular chaperone binds, and that binding triggers a conformational change which opens the structure. And in this open structure, the nascent polypeptide can fold in. So I'm going to show you a video of it. Remember that this is the holdase. It holds the polypeptide, it brings the polypeptide. This is the chaperone, and this is the one which assembles the polypeptide. So this is a video from the latest paper from Sheena Radford's group. This is the bacterial outer membrane. This is the bacterial inner membrane, and here is your chaperone. This is your nascent polypeptide coming in. Your holdase is bringing the protein, keeping it somehow in the unfolded state. And after it brings it in, it finds where the BAM complex is present, and it is supposed to hand it over to the BAM complex. So here is your BAM complex. Another holdase binds. Somehow it triggers a conformational change. This conformational change allows this polypeptide to now come in. And this coming in process, it's threaded in slowly through here, brought to this point. This gate acts as a template for the nascent polypeptide to grow. Once it is grown and once the structure, the folding is complete, then this protein, this holdase, leaves, and simultaneously the gate closes, and because the gate closes, this polypeptide is released into the membrane. Okay, so this was the mechanism that was recently proposed. Again, this is another hypothesis. There are only cryo-EM snapshots that are available for a few of these structures. So they put it all together to propose this as a complete model. It still remains a model. This is a cartoon. It's an animation. Yeah, generated from, so this is courtesy of the University of Leeds, as well as the team Phospho, which, let me pause this, which made this video.
Yeah,
They
quick
show.
question. Maybe you finish the sentence, you want to
They
speak?
have made fantastic other videos also.
Okay. Quick question. The question is, maybe this can be discussed at the end. This seems like a bimolecular process for SRA in some form, right? So is there an evidence for it? A kinetic evidence?
So how do you mean kinetic?
There are two molecules of SRA that are needed.
Yeah.
One that is going to have that loaded with the protein. The other one that is kind of, going around and then binding it, opening the protein for the transfer. So is there an evidence that you have two such surveys close by?
You have many such surveys. Some of them are associated with the polypeptide and some of them are
But
not.
in the transfer of the peptide, is there a kinetic evidence that is a square of the concentration of the survey?
No, there is no kinetic evidence yet. So, given all of this, we wanted to ask the most fundamental questions. How does BAM-A itself fold? Then, how does the folding, whether it agrees with or disagrees with Anfinson's hypothesis that the polypeptide carries the information to fold a protein, it doesn't need help. So how does that fit into this kind of a scenario? Second, what are the elements that are helping it stay stable and not stay stable? Third, how is the membrane helping in this process? And fourth, we asked the other questions from Kipling's Well-Serving Men, including how can we use this information for next generation therapeutics? And in our systems, in our mitochondria, we have the BAM homologue called SAM. So how are they similar? How are they different? And what does the SAM do then? And can we use all of this information to cure more relevant diseases and not just fight pathogens? So we started off by taking the BAM-A protein from E. coli, because that was the easiest organism to study. Here is a distribution of all of the tryptophans that are present. It has 13 tryptophans, very well enriched. So we were very happy. And we chose two different membrane systems, which had phosphatidyl ethanolamine or phosphatidyl glycerol, because these are the two lipids that are there in the bacterial outer membrane. So we wanted to see how does this contribute. And long story short, we made the protein in large quantities. So much so that students don't hesitate throwing a little bit of the protein down the drain. We managed to find a way to fold them into vesicles, and we checked that it was indeed folded by this electrophoretic mobility shift assay, where there is a difference in the migration of unboiled samples between the unfolded and the folded protein. Now, in addition to this, if you treat the unfolded protein with a strong protease like proteinase K, the whole thing gets digested. But if you treat it with the folded protein, it stays folded and stable. It's protease resistant. That was another way that we checked. Then we looked at the secondary structure content. We looked at TEM images to make sure that we saw the holes. And then finally, we also did the fluorescence experiments between the folded and the unfolded protein. Yes, all 5. Yes. Does it fold? It folds. Potra is not needed, even in the cell. Yeah, E. coli can work with just Potra 5. 1 through 4 can be removed. So, we carried out experiments where we looked at the thermodynamic stability, and there we did what is called as equilibrium titration measurements using tryptophan fluorescence, both in PE as well as in PG membranes, under a condition that gives us path independent folding, also called as no hysteresis condition. So with this, now that we had the system established, we made sure that we removed the cysteines because we didn't want any non-specific disulfide bonds, and we made a library of mutants where we systematically substituted every residue to alanine, got them checked, got them made, folded them, studied them, and put all of this information on what is called as a two-dimensional topology map. Now, what is shown in green is good, it's stabilizing. And what is shown in red is bad. It is intrinsically destabilizing the protein. And what you see is for the same protein, the PE membrane and the PG lipid do different things. Now, PE causes a larger number of residues to be destabilized. So the overall structure is intrinsically destabilized, particularly at the N-terminal zone, whereas in the case of phosphatidylglycerol, most of the barrel remains largely stable. Now, why this is important, we'll come to it in a minute, but lipid modulates the stability. The next thing that we did was kinetics experiments, where we followed the folding of this protein as a function of time. So this folding process again gave us an overall change in the kinetic free energy, from which we derived this value called as a phi value. A phi value compares the halfway process where the unfolded protein goes to the transition state structure and the full process change in free energy where the unfolded protein goes to the folded protein. So what is the contribution of every amino acid in this process? If we get a value of 1 or close to 1, then that residue folds soon. If we get a value close to 0, that residue folds late. And if we get a value close to 0.5, then maybe half the population is folded or all of the residues are halfway there. We additionally get values which are beyond 0 and 1. Those are the regions which make mistakes in the process of folding. So what did we do? We found out all of this in both the PE membrane as well as in the PG membrane, and we found 2 pathways, a minimum of 2 pathways that at least we were able to capture with our system. And this is a map for the fast pathway in PE, the slow pathway in PE, the fast pathway in PG, and the slow pathway in PG. So let's put it all together and compare. What you see in blue are the regions which are folded first, and you see a lot more blues in PG and lot fewer blues in PE. In fact, you have a lot more grays in PE. which means that the protein makes more mistakes in ethanolamine membranes compared to the glycerol membranes. Okay. So with all of this together, we were able to put together a model for how BAME assembles in the bacterial outer membrane when a pre-existing BAM is already there. So the Surrey SKP holdases, yeah.
It's very nice. Can you go back to the map?
Yeah.
So for some time in the protein folding community or the protein engineering community, when people used to have synthetic proteins fold into certain motifs, there was this idea that polar, non-polar combinations would go to a beta sheet. Okay. Do you see that? And the beta sheet would have this arrangement that hydrophilic and hydrophobic interfaces would be either be buried or seeing the water, right? So do you see, I can see there's alternate residues that are folding faster.
Yes.
Do you see the polar ones coming faster or the hydrophobic ones coming?
The hydrophobic
ones
come
are folding faster.
Yes, because
makes
we're
sense,
giving
right?
it in the membrane. So the hydrophobic ones.
Okay,
are
that makes sense. So
Yeah.
because they're exposed in such a way to the
Yes.
membrane. Okay,
Yes.
good. Thank you.
So with this, we were able to put together a mechanism with which we think BAME folds. So it starts off maybe in the cell by attaching to one of these holdase molecules, and then it is handed over to an existing BAME with its POTRA domain. The POTRA collects this polypeptide from the holdase like we saw in the video, and then after that the holdase, in our case we assume that it goes away, but maybe it stays, we don't know. But once it is handed over, then comes the choice. The choice is it can either go through what I will explain in D or in E based on what the membrane is. Now, if the membrane is phosphatidyl ethanolamine, then what you see is the phosphatidyl ethanolamine actively prevents the polypeptide from spontaneously popping in, which means that your existing BAM complex has to rope it in one hairpin at a time. That's how it folds. So
this you have this liposome or whatever it's
Yes.
called, and you already have a BAME inside it, is it?
we don't. In our experiments, we don't. We are doing it in an unassisted folding manner.
So in your experiments, there is no
is
this
no place.
POTRA, this, that, nothing is there.
This blue one is not there. What we have is this maroon one.
But your model actually says includes all of this other stuff.
Yes, because the way it folds in the bacterial outer membrane is it requires a pre-existing BAM. Otherwise, it won't insert because there are mechanisms available to stop it from inserting.
No, I understand. But then, meaning, so the experiment that you're doing is going to show you something, and you're assuming that what is happening in your experiment is the same as what will happen in bacteria with this BAME present.
Yes,
Okay.
because what we are doing when we do the experiments, the amount of ethanolamine or the amount of glycerol that we add in the lipids in our membranes is lesser. Whereas what is there in the bacterial outer membrane is 75% PE and 25% to 30% PG.
No, I know.
So-
I'm just saying that you are assuming that the BAME being in the bilayer does not somehow change the mechanism of folding,
Yes,
right? So that's
Yes.
what you're assuming. Okay,
yes.
fine.
So when you have PE in the membrane, the ethanolamine is there in the membrane, then what happens is your incoming nascent BAME folds one hairpin at a time and slowly inserts itself, so that it slowly starts elongating. And the evidence for that we have is hairpins that are formed in our experiments without the entire barrel being formed, and these hairpins are actually stuck. They are not pushed to the completely folded state immediately. So these hairpin structures, 11 and 12 hairpin, then 5 through 8, and then 10 through 15, they are all formed, but they are not fully inserted to form the barrel. They are stuck. Why? Because PE, the ethanolamine, stops it from folding instantaneously. On the other hand, so this the previous one goes through the beta augmentation model that was proposed, hypothesized before for another system. On the other hand, when you have the phosphatidylglycerol as your lipid, then the folding pathway is very different. Here what happens is the polypeptide starts folding even before it goes into the membrane. So the folding over here can happen on the surface of the membrane, and then akin to the swing mechanism, the whole segment inserts itself. And this insertion we see because a large segment of the protein is already folded even before the entire barrel is fully made. Whereas in the case of PE, we only have short segments that were folded. Here, larger segments are folded, which is why we are certain that the polypeptide follows two different mechanisms based on which lipid it is. Now, once this decision is crossed, then the subsequent processes are rather straightforward, where the nascent polypeptide, nascent BAME is fully folded, released into the membrane, and then it finds its other auxiliary binding partners and forms a BAM complex, which is now ready to fold a new polypeptide. So this is the mechanism we think happens in the case of BAME. Now, why is this really important? In the case of PE, the dynamics is enhanced, whereas in the case of PG, stability is enhanced. That is, PE hinders folding, whereas the glycerol enhances the folding process. PE, on the other hand, enhances function, whereas PG suppresses function. Why is that? Because in the case of PE, the barrel is metastable, and something that is metastable will have higher dynamicity. It won't be a rigid entity. PG, on the other hand, makes BAME a rigid entity, and a rigid entity cannot do this open-close dynamics that it has to do in order to bring in a nascent polypeptide. So the dynamics plays a role in the function of this protein. And additional evidence for that comes from other studies which looked at the localization. For instance, if you look at a substrate OMP, you see that it is organized in patches and more towards the center of the cell, whereas if you look at the BAM complex, it is distributed throughout, even at the edges of the cell. So the BAM complex additionally reaches out to your lipopolysaccharide and forms an interaction with it when the polysaccharide is being made, but when it is mature, it loses that interaction. So place of the cell which is rapidly dividing versus a place of a cell which is already divided and aged will now have two different features. And that is what our data shows. So in the case of phosphatidylethanolamine, which is enriched from this part to this part of the bacterium, where the bacterium is rapidly becoming longer and longer, that part requires new BAM complex because it needs to grow, it needs to put in new proteins, it needs to put in other OMPs, so the BAM complex has to be functional. But it is less tolerant to mistakes. So PE suppresses the folding so that whatever nascent polypeptide comes is folded correctly and there are no mistakes. On the other hand, when you have phosphatidylglycerol, that is largely found at the edges of the bacterium. And remember, when a bacterium grows, the outermost bacterium or bacteria on the two edges of the line are the oldest. The ones inside are the youngest. So there is a demarcation there as well. And the edges are the ones which have the oldest proteins dumped there, because you can't pull them out and chop them away. They'll just be dumped into the environment or put in as discard. So it's that junk room which is there somewhere in this building and nobody talks about it. So that similar thing happens in the case of bacteria, and that part is enriched with phosphatidylglycerol. And phosphatidylglycerol helps BAME fold but doesn't help it function. So what the bacterium does is it makes sure that whichever one is folded all right, but the new polypeptide doesn't go there. It goes into the region that is actively dividing. It makes sense, right? Wherever there is active division, you need more protein. And we were able to confirm this also with all atom MD, where we see that irrespective of the POTRA, in the case of glycerol, the dynamics don't change much, but with the POTRA and in the presence of PE, there is greater dynamics in the barrel itself. So that confirms what we are proposing between PE and PG based differences in the function. So we started off with 4 questions. How does it fold? It can fold on its own, but the membrane plays a role. The stabilizing elements are differentially distributed. It's not residue specific, it's position specific. N-terminal zone versus the C-terminal zone. Then the membrane plays a very important role. If you have glycerol, it folds, keeps it stable, doesn't allow it to do this open-close function. Whereas if it is in ethanolamine, it makes sure that it's folding slowly, correctly, and gives it the room to do the dynamics. So then what region do we target? What we find is, unlike loop 6, which was proposed earlier in the literature, loop 4 is what is essential for this entire process to happen. So maybe loop 4 is what we should target for the next generation therapeutics. And I also said that SAM is what we have in mitochondria. So what are the differences and how do we use it for other diseases? So we looked at SAM50. This is a core protein of the sorting and assembly machinery. Just like how BAM-A is for BAM, SAM50 is for SAM. And we studied this protein by looking at its stability, whether it's folded, whether it has secondary structure content. Unlike BAM-A, this particular protein shows gating characteristics. So we also did single channel electrophysiology to check that the gating is there. And since we were able to complement it with yeast mitochondrial SAM, we were able to show that it is fully functional. We did the same experiments, map the stability. This particular barrel is more destabilized than your BAM-A, and the dynamics changes based on the mutations. We map this function as well as the folding pathway. And what we see is over here, it's a mix of both the beta augmentation as well as the swing, because a part of the protein folds before, but it waits for other segments to assemble first before the barrel is inserted and it is released into the membrane. So the mechanism of folding of the eukaryotic homologue of the bacterium is different. It follows different mechanisms. So here again, we asked multiple questions. We were able to show that not just for this particular protein SAM, but also the other beta barrels that are there in the mitochondrial outer membrane, VDAC and TOM40, we were able to identify how the protein folds in each case. It's unique for each barrel. The stabilizing factors are different based on what function it does. The functional residues, arginines, lysines, aspartates, and glutamates, differentially distributed. Cysteines are unusually enriched in specific cases. They act as Ross sensors, and they also are important for importing other proteins. And finally, we were also able to map the regions which allow these proteins to talk to each other and form an interaction network in the membrane itself. Now that we have all of this information, our next question is, where does this lead us in terms of diseases? What we want to see is, how does this one molecule alpha-synuclein get into the mitochondria? Does it take the pathway through VDAC? Does it take the pathway through TOM? Does it use SAM? How does it get it? Because it has no business in the mitochondrion, but it gets there. So we want to see how it gets there so that we can help alleviate conditions related to neurodegeneration. So we have been able to map some binding interaction interfaces. We also made a series of a library of more than, I think almost 60 peptides, which are good for oligomerization and aggregation inhibition or promotion. And what we are doing now is to test to see which of these deplete the protein from aggregating, suppress aggregation, so that we can use it for neurodegeneration, and which are those proteins, which are those peptides which facilitate the aggregation process, and maybe we could use it somehow to help alleviate cancer by triggering cell death. So with this, I'd like to thank my group. Shashank, Gifty and Tanya were the primary people who initiated the work on VDAC. All of the BAM work that I presented today was done by Anjana. Rashika did all of the SAM work that I presented today. And Udit, Adish, and Kinjal are the ones who are looking at how synuclein gets in through different proteins. Dipshika is looking at how mitochondria talk to endoplasmic reticulum. I also like to thank my collaborator, Professor Vikas Jain, for helping with all the MD simulations. Then my funding agencies who have been very generous, very timely in releasing the money. Of course, there was some amount of begging, threatening that was done, but the money came in through. And so the India Alliance has been the major contributor, followed by SERB as well as the Department of Biotechnology. And last but not the least, I like to thank my favorite amino acid, tryptophan. I fell in love with it in 2000. I used to hate bananas, but now I love bananas because bananas have tryptophan. So tryptophan for being the most faithful reporter. And thank you all for your patience.
Thank you, Mahalakshmi. That was very nice. So, questions for, yes.
Hi, Mahalakshmi. Excellent work.
Thank you.
As you presented, this is a very complex problem, especially folding of membrane proteins. I work on soluble proteins. They themselves are really complex. But especially because it's so complex process, either folding by themselves or through chaperones, even could be even more complex.
Yes.
So there might be a lot of intermediates, multiple
Yes.
parallel pathways.
Absolutely.
So how would you actually, put together? Because of course you have given the overall picture from a lot of studies, but how would actually one begin to think about this complex energy landscape of these proteins where there might be many intermediates, or it could be even misfolding?
Yes.
So how would you actually address such complex issues in
The only thing we need is time, because the technology hasn't caught up to the extent that it has for other systems. So with time, I think we'll be able to, not just us, by working as an international collaboration to look at different aspects, someday we'll be able to capture each and every process. Like I said, we only were able to capture 2 pathways, but I am very certain that there are at least 100 different ways in which it can fold. And we were only able to get 2. So there are multiple ways that a protein can assemble, even with the chaperone's assistance. We just don't have the tools yet.
Another short question. So what is roughly the time scale of folding of membrane proteins compared to soluble systems?
Micro to milliseconds.
Very slow, right? Compared to
slow.
Thank you.
Okay, could you go with this tool?
When you talk about SAM, you said that a part of protein is folded before it goes into the membrane.
Yes.
For BAM, most of the protein occurs once it goes into the membrane. So how did you do it? Can it not be possible that BAM also folded the same similar way, that some of it folded earlier and then released into the membrane? How did you think of it?
So it depends on which part of the strand was folded first, which part folded second. What I didn't delve into were which residues were blue in that topology diagram. So if something that is going to go to the top is blue first, and the rest of it is still red, that means half of it is inside. But if the bottom part and the top part are blue, that means that the entire strand is formed. And that can be formed when it is sitting in the membrane already. So that way we were able to demarcate which region was folded, which was not, which was getting there, and so on.
Was it because of the difference in the environment of SAM and BAM or the sequence?
I believe it's all in the primary sequence. So what we have done now is we have hybridized them.
What do you mean by hybridized?
Oh, we took a part of BAM, we took a part of SAM, and we now made a hybrid.
Okay. Sure. Okay. Questions? Can I ask a question and then I give it to you? No, please. So this entire thing that looks that you showed the video of, that actually is intriguing to many of us, because
Well, it's not mine.
hardly,
Sheena.
yes, Sheena Radford's
Radford,
group, whatever that. But these prevailing movies are built on some models and some experiments, right? Some hypothesis. So, in the case where, do you have any evidence, or you or anybody in the field has an evidence that once that SIR A binds without, that's empty SIR A, binds, you have this opening up and there is another SIR A that has to come to transfer. Is there an evidence for a ternary complex like that?
So this evidence came just now, and that was that NatCom paper which came from Sheena's
So
group.
there is an evidence of this ternary complex.
Cryo-EM. That's the evidence. Before that, there was no direct evidence that an empty SIR A binds to BAM. That evidence was not there. Well, if
yeah.
they would have used at least an 8-stranded structure as a template, right? And that is 150 amino acids. So you suspect that it's there, but maybe.
Okay.
It's possible. Anything is possible. मैं देख रही हूँ और किसी के पास question है क्या? So I like these Potra domains. If you just chop off the Pame and let it go, and do the Potra fold by themselves?
Yes, they fold by themselves.
Okay. Do they?
Each domain folds on its own also.
No, but if you put them all together, then they fold by themselves.
Yes, they fold by
Do
themselves.
they bind a polypeptide chain by themselves?
They can also bind a polypeptide
So
chain.
why are you not using your own Potra to in this particular mechanism? Because it seems like you have the Potra.
Yeah, so I have the Potra.
I know, but they should, I mean, I would think that they fold by themselves.
Yeah.
Or onto this unfolded rest of Bame.
Okay.
And then, meaning so they sort of, it's self-chaperoned by its own Potra, which is folded. And then you insert. That's
Okay.
how I would, I would think it happen. I would imagine.
So even if it happens that way, that would be one of the earliest events. And in that earliest event, we see 2-3 strands that are folded. Perhaps that is Potra bound.
All right, but have you done a 5-value? So, okay, so we'll come back to the 5-value, but have you done a 5-value of just the Potra? Do you know if...
No. We were more focused only on the TM.
Have you done 5-value of the Potra at all?
No, not for the BAME. For the human SAM50, we have the 5-value of the Potra, of the TMD, everything.
Okay.
But we are doing the analysis. So maybe in a couple of months, I'll have the answers for you.
Thank you. We will have a chance to talk to her.
So you mentioned about PE and PG, but
Yes.
you didn't talk about the acyl chain length, because 14, which you have used,
Yes.
is not very common in
Yes,
outer
it is
membrane.
not very common.
So what happens if you use 16, which is more?
The process is slower. So we tried that. We didn't try that for BAM-A, because for BAM-A, even getting it to fold, we had to do a lot of tricks to get the process happening. And it went to the point that even Sheena was amazed that how did we get it, and how did we get it for so many mutants. And I told her there is just one key ingredient, and that key ingredient is Anjana. You wait for her to graduate and take her.
So what about cardiolipin?
So
is it there?
these, cardiolipin, 16 carbon, changing the head group and changing the saturation of the chain, we have addressed all of that for the mitochondrial parents. And there, the folding process gets slower, and the addition of cardiolipin changes its function, but not its folding.
So you mentioned about E. coli cell dividing and so on. Is there a segregation of PE and PG in there?
Yes, PE is more towards the middle, PG is more towards the edges. There is PE at the edges, there is PG in the middle also, but the relative proportions are enrichment of PG towards the edges.
Okay. Well, I think we have Mahalakshmi for two more sessions of this meeting, as well as tomorrow. So let's thank Mahalakshmi once again with a big round of applause.
Thank you all.
And before I end formally the proceedings, I have to, by definition, I have to give Mahalakshmi because Kalaivanan has brought a gift for you. Kalaivanan is not here. I was
Okay.
talking to the empty seat, but but Kalaivanan brought a gift for you to take home, and it's a beautiful book about Homi Bhabha and all the paintings and the archives. So if you gladly take it home, it will be. Yeah, I think that's
Okay.
why we have to do this. Okay.
Thank
And
you.
there is something which is also there, which is sort of something you can use. It's a useful writing pad.
Oh, okay. Thank you. I love stationery. I joke about this with my students. They can take my kidney, both of it, but they don't dare touch my pen.
Okay. Thank you.
Thank
So
you.
let's walk towards the West Canteen. Actually, outside the West Canteen, we have some snacks and we can have more discussions with Mahalakshmi. Thank you. Thank you to all the audiovisual people for making it happen. Thank you.